Good morning and good afternoon. Welcome to our PerkinElmer webinar on more predictive results in cell-based assays, combining target-based and phenotypic approaches by using our new EnSight Multimode Plate Reader. Before we get started, let me introduce the speakers. I'm Volker Eckelt, Portfolio Director for Multimode Detection at PerkinElmer and based in Hamburg in Germany. And I will give you the introduction. The second part will be covered by Norbert Garbow. He's our principal application scientist based in Hamburg as well, and he will give you an introduction in cell-based imaging. Last but not least, our guest from the ScreeningPort, Phil Gribbon. Phil is head of screening Fraunhofer IME at the ScreeningPort in Hamburg, and he will talk about assay development, anti-cancer assays. So welcome, and let's get started. So what will you learn in this seminar? First of all, combining cell imaging with labeled and label-free technologies to get more predictive data earlier in the drug discovery process, to improve your data quality and reduce cost in cell-based assaying using the new EnSight Multimode Plate Reader, and you will learn how you can use the cell imaging capabilities in anti-cancer drug discovery. So PerkinElmer gives a lot of tools, instrumentation, reagents, software to develop your translational research. And now I like to focus on the early steps in the workflow in order to help you to reduce cost per data point and to improve clinical relevance on the other side. So the EnSight Multimode Reader we are launching now. We are very happy and proud. So what is the EnSight? What do you get? First of all, it's the only benchtop system which offers multimode detection, alpha, label-free, and imaging. You have a truly orthogonal approach here. You can run target-based, phenotypic, or the combination of both assays on the machine to gain more predictive results. Last but not least, you have a new image-based cytometry module on board, which gives you 4 colors, bright field, digital phase imaging at high speed. When we talk about high speed, we talk about 4 minutes per color to read a 384 well plate. So what kind of assays can you run on the new machine? Let me introduce the overall portfolio, and then you will see what you can do with the EnSight. So for the multimode detection, we have for the biochemical assays the easy reader. When you want to run cell-based assays, integrated assays, we talk about numbers or number per well. You can use the Victor, our Envision, or for ultra-high throughput screening, the ViewLux. Some scientists are more interested in cell imaging, subcellular, and here we offer the Operator. And what we have recently launched is the Opera Phoenix. Not really covered is the space in between the cytometry market. And now with the imaging capability of the EnSight, we fill this gap and build the bridge. So what can the EnSight offer to the multimode detection world? First of all, as I pointed out, you have a truly orthogonal approach. All conventional MMD technologies plus label-free can be combined with cell imaging in one workflow. You have a fast imaging cytometry module on board. All technologies are field upgradable, and you have a nice system for the mid-range market. What can the EnSight offer for the high content world? First of all, if you are interested in simple assays at cellular resolution, you get access to the conventional MMD technologies like alpha, label-free, or time-resolved fluorescence. It's easy to use and no special training is required. And the system would free up your high content instrumentation if you use it upfront. And it can help with any budget or space restrictions. So in summary, what is the plate reader? You get new software, our new Kaleido software for data acquisition and analysis. This is a workflow, and I'll show you on the next slide how it looks like. New instrument control and data structure, and we are able to provide you customized data evaluation. We have a new hardware on board. You see a new look and feel, new technologies on board, and all technologies are feed upgradable. The cell imaging is made for single cell resolution, for example, cell counting, fast 96-384 well plate reading, and you can combine it with conventional MND technologies. So here's a first draft of features and benefits you get with the instrumentation. Fast and flexible, powerful software. This is workflow-based for high performance and support. And Acapella, our well-known software for high content, is running in the background as engine for data analysis. It's modular and designed to upgrade all technologies. You get the system what you need now. It's customized, tailored, and later on you can upgrade the system according to your needs, so you're ready for the future. We integrated well imaging on board. That gives you image-based fluorescence for labeled multicolor assays. You have access to 2 stainless modes like bright field and digital phase contrast. You can use this for increased QC in your cell-based assays, for example, cell counting. We have made a special optical design which includes laser-based autofocus and a scientific CMOS camera. With this, you gain high performance at low light and high speed. I've mentioned already Kaleido and our pre-written data analysis. Here you leverage our over 10 years experiences in high content, and that allows you to run in a simple and fast way our known high content algorithms. Last but not least, with this system, you have access to broad range of application, multimode detection, label-free and imaging for multiplexing and orthogonal workflows. So here you see our Kaleido new software for acquisition and data analysis. It's a workflow based. So on the top here, you set up a protocol, run a protocol, reanalyze, and so forth. On the left side, you determine which technology you want to use in your protocol. For example, alpha, TRF, the imaging, and so forth. On the right side, you find the plate map. Here you can determine the number of positive, negative controls, standard curves, and so forth. And in the centrum, you have the content area. Here you see the results, the graphics. So in summary, quick and easy, you can analyze and reanalyze your data. Either you use the instrument PC or you can do it in your office PC. You get your data as list, graph, or heat map, or whatever is required. What kind of technologies are on board? So here you see in this graph, we have absorbance, fluorescence, time-resolved fluorescence, ultra-sensitive luminescence, alpha, of course, epic label-free technology, and now the well imaging. And that enables you to run different kinds of assays. You can run our alpha assays on board. You can run different fluorescence assays, absorbance, lens or DeFia assays, luminescence assays, and of course, epic label-free assays. What is new now is image-based cytometry for fluorescence, bright field, and digital phase contrast. Now I'd like to come a step closer to our new imaging module. So on board we have 4 LEDs in the UV range, blue range, green and red excitation. We have two transmission modes, meaning bright field and digital phase imaging. We have a fast laser autofocus on board, which helps us to have very quick reading times. Here you find our CMOS camera, and the CMOS camera helps us to have high performance at low light and high speed. This is a special optical design. You will get more details from Norbert in the next part of our presentation. And of course, you can combine the imaging modules with our conventional multimode detection. With this, I'd like to show you different applications, and I'm very sure we get a longer list. So cell counting is one, normalization, tox assays, you get examples in this webinar, cell migration, cell signaling, phenotypic assays, as well as clone selection. So all this type of assays you can run at cellular resolution in an endpoint or kinetic mode for live or fixed cells. You can combine it in one workflow, and that really leads you to the holy grail, to orthogonal confirmation. And with this, I'd like to hand over to Norbert. Oops. And he will give you more details on the imaging module.
Okay. Thank you, Volker. As already was pointed out, the EnSight does support all the multimode detection technologies that are well known from former instruments and from the Envision. What we added now is the imaging capability, and I would like to give you some examples on this capability. To start with, set up a few details on the technology. We are going for single cell resolution, focusing on better statistics using multiple cells, or many cells in our field of view. So we did set up the optics in a way that we imaged approximately 3 microns of the sample to 1 pixel on the camera. This gives us the freedom to have a very large field of view, which easily covers the full valve in a 384 valve format, and even covers almost a complete valve in a 96 valve plate. We have up to 4 different fluorescence channels, plus the non-stain methods, bright field and digital phase imaging. We know that image evaluation, particularly automated image evaluation, can become a very interesting and sometimes difficult story. So starting with our algorithms developed in the high content world, we did invest quite a bit of effort to simplify the image evaluation tasks for our customers, for the users. So we have a broad range of applications that is already covered by the available algorithm and methods for the evaluation, and this selection of algorithms will increase and grow over time. We are also capable to establish custom-tailored evaluation solutions if required. You have seen the sketch already. Basically, the imaging module consists of the sample. That's not part of the imaging module. We use a 4-fold objective lens. We have several diodes for excitation of fluorescence. We have a multi-band path optics to separate between excitation light and the fluorescence emitting from the sample. And we have a scientific CMOS camera for highly efficient detection of fluorescence. When we are going to excite the sample using, for example, the UV diode, we just have to switch on one of the fluorescent diodes. Where is the? I can't switch here, sorry. So we just have to switch on one of the fluorescent diodes to excite the sample, and the emitted fluorescence light from the sample will be collected by the objective lens, pass through the multi-band detection optics, the multi-wavelength band detection optics to the scientific CMOS camera. The advantage of this approach is that we only have to switch between different excitation wavelengths to use multicolor assays. So switching from a single color assay to a dual color assay does not require twice of the measurement time, but just at the exposure time required for the second color. This has a benefit that we use about 4 minutes for a single color in 384 well plates, and only require about 5 minutes for the second color, so 1 additional minute in this case. As an example, I here have a dual color overlay of cells which have been stained with a nuclear stain and a cytoplasm stain. There are 2 individual channels which are both displayed in black and white here on the left side, and the overlay is a color image for 2 colors, in this case blue and green. Of course, it's easy to add a third or even fourth color. My personal favorite, however, is currently bright field imaging. While the fluorescence channels can establish very specific assays, the bright field imaging has the benefit of not relying on fluorescent stains. So even without any fluorescent stain, we can image cells and characterize them. If we have a fluorescent stain in addition, we can combine it as we want. There's no interference between both methods. So we use a Phipps diode on the top of the sample carrier to illuminate the sample. The illuminated sample will then be imaged by the objective lens onto the scientific CMOS camera. And here we have 2 typical images of these measurement modes. On the left side for a 384 well plate, on the right side for a 96 well format. If you look carefully, you can see on the left image that you, at the very rim of the display, already can see the next well of the plate. What you also see is that there is a significant change in the intensity in this measurement mode. This is due to the liquid surface interface, which acts as a strong lens. While this distorts the pure intensity display in bright field, it doesn't distort the evaluation. So this is not an issue with the evaluation of these images. These 2 images are the only images in this talk that show the full field of view. The subsequent images have been cut down to the size of a well or to a detail in the well, just for clarity of display. And here you can see a comparison of the 2 stainless imaging modes. On the left side, the classical bright field image. You can see to some extent the individual cells as changing intensity in bright field. And on the right side, you can see the digital face image, which actually is derived from bright field images. We use dedicated algorithm to calculate from bright field images this face image. This is possible also for microreader plates. Some of you may be aware from microscopy that phase contrast microscopy requires special optics and is sometimes a little tedious to align. In this case, we use numerical method, which do not require any additional optical elements or a complicated setup of the measurement. The digital phase images at the final step of evaluation look very similar to a simple nuclear stain. They are not exactly, but they still can nicely be used for segmentation of individual cells. So this method is capable to count individual cells without any stain. We'll see some examples for evaluation of the simpler bright field images later on. Here's a list of applications we did test on the EnSight recently. We had some applications in the range in the area of quality control, and we had some tests combining fluorescence measurement with stainless measurements. I'm going to focus on a simple comparison of several methods using for cell counting with and without staining. I will continue with the main application for quality control in cellular assays, and I will give a short example on live dead assay later on. In this figure, you see on the left side cells of the R431 cell type, which tend to grow in patches, which have been stained with Höchst as nuclear stain. On the right side, we have also stained cells in stained HeLa cells. You can see that the HeLa cells go quite alone, so they are well spread out over the whole plate, while the A431 cells are growing in patches. Both can easily be evaluated and segmented by our algorithm for single cell counting. Please note the bow-shaped cell group in the center of the left image. We will find this again in later images. We can use these images, and those are the very same images as before, for cell counting in the nuclear channel. We also can use the bright field images to determine the degree of confluency, which is shown in larger displays in the lower part. And here we see, as you might estimate by eye, that for the A431 cells, the confluency is a bit lower than for the HeLa cells. We can do the same story with digital phase imaging, shown in the top display. And the left display also shows the cell segmentation information here. For comparison, in the lower display, the nuclear stain is given. So we did compare cell counting or determination of cell density in different methods. Of course, for cell seeding, we did use a separate instrument, a cell counter, to estimate the number of seeded cells. We used nuclear staining for cell counting, which is kind of a gold standard for this kind of application. We use digital phase imaging, which also counts cells, but without the need of any stain. And we determine the degree of confluency from bright field images. Here we just get the area covered by cells, but not a cell number. But we also avoid any fluorescent stain. All methods correlate nicely in a large range of cell densities. For later part, we need a bit more detail on the way the degree of confluency is calculated. What you can see here in this detailed view are cells spread out in the center of a valve. In the area covered by cells, we see some fluctuation of the intensity, which basically follows the shape of the individual cells. In the area not covered by cells, the intensity is very smooth, so there are no major changes in intensity, except if there's a dust particle, for example. And actually, this texture structure is evaluated by the software that calculates the confluency. In the bottom display, you should see the texture parameter we call roughness that characterizes the intensity fluctuations in the area covered by the cells, but also in the area not covered by the cells, where it is much lower. When calculating the degree of confluency, you of course get the basic parameter, which percentage of the well is covered by cells, but we get additional information on the texture of the cells, like the roughness in the foreground, so within the area covered by cells, the roughness in the background, and the range of roughness or roughness factor that compares the highest value of the texture to the lowest value of the texture, which sometimes gives surprisingly important add-on information for certain samples. What can we do with confluency analysis? Of course, the basic and most important thing is to confirm the quality of the cells in your samples. Here we have a typical picture of cell culture that may happen during processing. The left display shows a well-behaved well. The center and right displays are somewhat disturbed. On the right side, the tip of the pipette did scratch the bottom. On the left side, during cell seeding, a bubble disturbed the distribution of the cells. So the degree of constituency is lower here. In this display, on the left side, using MCF7 cells, we have a very healthy structure. On the left side, there are cells adherent to the sample carrier with some rounded up cells in between, and we have empty areas with no cells and a very smooth intensity distribution. On the right side, we found a well that has been contaminated by bacteria. In this case, the cells have been destroyed. They are just gray bubbles, and the area in the background without cells is covered by bacteria, which does increase the texture here. So the range of the texture, the roughness factor as we call it, is reduced dramatically in this case, which enables automatic detection of such effects. Can we use it for something else? Yes, of course. As all of you are aware of, adherent cells that are treated with toxic substances will shrink and round up. And in this display, we have this as example for HeLa cells. On the left side, the cells have been incubated overnight without DMSO, so they extend their attaching to the well and use quite a large area of the plate of the well for their growth. On the right side, the cell number of cells has been treated with 4% DMSO, which is already a toxic concentration. And here you see that the cells are separated. They are very small and very rich in contrast. So the toxic effects in this case affect both the cell coverage as well as the texture. We can do this in more detail. And as we could compare the cell status prior treatment and after treatment, we can calculate the difference between both. We see in the left display that the degree of confluency for low DMSO concentrations did increase by something like 4 to 8%, which means when we had 35% confluency prior to the treatment, we have now 39 to 44%. With higher concentrations of DMSO, of course, the cells to shrink or detach and round up, so the degree of confluency is reduced. As you can see here, if you look closer, for a moderate concentration of DMSO, the cell coverage increases even more, so these cells seem to like a bit of DMSO. With the roughness shown on the right side, we see that the roughness parameter during the incubation has been reduced by some numbers, something like -10, which indicates that the initial cells have been affected by some dead cells. The dead cells did not continue to grow, but the live cells did continue to grow and have a larger percentage, a larger effect on the roughness. With higher toxicity, the roughness parameter does increase significantly, and the small error bars indicate that this is a very robust parameter for evaluation. So I did try to show you that even the basic evaluations without using any fluorescent stain are very valuable for many applications in the cellular area. I did not show you details on digital phase images combined with fluorescence, e.g. for analysis of transfection rates. And I did also not show up the examples we already have for migration assays that will come up a bit later. With this, I would like to hand over to Phil for the next presentation. Thank you.
Okay. Before we get started, I just wanted to tell you that we have a Q&A field, so if any questions in between, please type it in, and at the end we have blocked 10 minutes for a Q&A session. And now we like to continue with Phil's talk.
Okay. Thanks very much, Norbert and Volker. Talk. Okay, so I'm Phil Gribbon from Fraunhofer IME here in Hamburg, formerly known as ScreeningPort. Today I want to talk about how we've been working with the PerkinElmer team to see how we can use the imaging capability of the EnSight to determine cell viability and also cell growth effects and cell cytotoxicity effects. Typically, we normally use ATP detection within our lab, and we'll show you some results from our existing processes. But now we want to see how we can use the bright field facility offered by the EnSight as a supplement to that, and eventually maybe we can use it to replace that as a phenotypic readout of cell viability. So just a quick note on the ScreeningPort and the IME. Our main activity is involved in small molecule drug discovery. We have 500,000 compounds in our library, a number of automated screening systems, including some from PerkinElmer. We work across several indication areas, particularly in neurodegenerative diseases, cardiovascular diseases, but also we have several projects in the cancer area where we apply these phenotypic methods to identify compound activities. We have a translational group which develops molecular biomarkers, and we support a number of phase 2 clinical trials. We also get involved in bioinformatics, and we work very closely here in Hamburg with the DAISY X-ray source here on target-based structural biology programs. I think what's also very important for us is our enabling technology activities, where we work in close partnership with organizations like PerkinElmer to try and work with them to develop and benchmark novel assays. And this is where this is really the result of a very profitable and intensive collaboration with PerkinElmer over the last few months, and looking at the early versions of the instrumentation. Obviously, it's by no means a complete study. It's really a view of what the possibilities that will be offered by this technique are. And going to the next stage. Okay, so I just want to point out what we're normally doing in terms of our phenotypic cell viability assays. We really use the Promega cell titer grow methodology, whereby the measurement of the ATP concentration is used as a surrogate for the number of cells and a surrogate for the cell viability. So the more cells you have, the more ATP you have, the higher signal you have produced by the luciferase reporter in there. And this really is a very flexible technique in our hands, certainly. It allows miniaturization, and you can get your assay volume down to about 20 microliters, and even be looking at the range of several hundred cells per well. So up till now, I think ourselves and I think a large number of people we work with have been very happy with this technology, and it works well. But it isn't like all assay methods; it isn't without some drawbacks. And here's just a sort of an overview of some data that we've shown where we've used this technique. It's out in a recent paper by my colleagues Bernard Evinger. So that obviously the first thing that you'd like to do when you're validating these cell technical assays is to confirm that it's not too dependent upon DMSO concentration. So here you can see, similar to what the data shown by Norbert, that you see you get a reduction in cell viability, increasing DMSO concentration. Then you want to be able to look at the linearity of the response with respect to the number of cells, and you can see that it's extremely linear. And then the third point that you would get involved with in any assay validation is to look at the response of standard compounds. Here is an M14 melanoma cell line that we're using, and you can see that we get reasonable dose responses, and they're comparable for Cisplatin and 6 mercaptopurins, Doraspor, and paclitaxel standard compounds. They're comparable with what you can see in the literature. I think what is quite interesting and is sort of indicative of this and pretty much any other method is that there's always a potential for compounds to interfere with the detection. So typically within our process, we're able to run a counter assay. And in the counter assay methodology, the idea is that at the very short time points, at time 0, you measure the response of the system just to the presence of the compound, and then you can also compare that to the value at 72 hours. So here for cisplatin, for instance, we can see the toxicity of the compound is what you expect at 72 hours. But at much shorter time points, and here it's in the gray line, you can see you also get an effect. I'll make that a bit more clear. The gray line here, you can see you also get a potential what looks like a cytotoxic effect. So this is just really an indication that, as with any methodology, your compound here can be interfering with your report system. In this case, it's probably interfering with the receptors. And those are the types of features that you want to really avoid, particularly in phenotypic assays, particularly where your compounds may be relatively weak. So we're doing phenotypic assays, but just as a little bit of background to it, we're actually very interested in being able to move from a phenotypic observation through to some information about what the underlying target is. And when we're using the NCI panel, and we have the 60 cell lines of that, I think we've developed 35 assays so far. When we're using that panel, we can actually take the data set, the profile of activities against each of those different cell lines, and then use that to compare against profiles for established compounds with established mechanisms of action. For instance, kinase inhibitors or HDAC inhibitors or DNA cross-linking compounds. And you can use those bioinformatics tools. And here's an example of it actually does work here. So you can see we're screening a particular compound which is active against cells which express the VEGF receptor 2. And in this case, only these RPMI8226 cells express that. And you can see that there's a differential level of cytotoxicity for these cells compared to cell lines which don't express the same receptor. So there is an opportunity within these phenotypic assays and using these cell lines to identify compounds which have target-specific effects. How can we sort of see this in a large-scale analysis? Well, here's an example where we've taken cisplatin results and we've compared the profile over 35 cell lines. And you can see here at the top panel, we're comparing against a set of 171 compounds on the standard data set within the compare analysis. And you can see that cisplatin-like, and cisplatin's mechanism of action is to actually cross-link DNA. Cisplatin-like compounds appear within the most highly ranked compounds, including cisplatin itself at number 4. You can compare this cisplatin data with what's happening against a diversity set of 38,000 compounds, and you can see again that cisplatin-like compounds appear in there. So we've got using these classical techniques like ATP determination, profiling multiple cell lines, you're able to get to a point where you can actually potentially identify compounds with interesting mechanisms of action, and then can use that later on to select the compounds which you want to progress within the program. But that's not all, and I think we need to understand how we can improve the process. ATP detection as a methodology is certainly not a universal readout. And it's certainly the case that some cell growth and toxicity mechanisms are not adequately affected. And for those who are interested, I would certainly direct you to a recent publication in +1 by Chan et al., which shows that compounds which are able to actually increase the size of cells and also the mitochondrial cells. And these are typically DNA synthesis targeting agents such as gemcitabine and etoposide. You actually you're underestimating the potency and efficacy of these compounds which can cause these changes in the cells, particularly in increasing the mitochondrial content. Also, one of the problems with ATP determination, as with any other endpoint assay, is that you get to a point where you're only really able to take one snapshot of the assay itself, and it's difficult to be able to do kinetic evaluations. And that's potentially very interesting information to have. It's certainly the case that the mechanism of action, for instance, membrane disrupting agents, is very quick, and you see a rapid onset of cytotoxicity, whereas those cisplatin who are involved in DNA cross-linking, their kinetics are much slower, and you only really see effect in tens of hours or days. So certainly in our case and programs we've been involved with, having that additional kinetic information about when compounds affect cell growth, when compounds are cytotoxic, is also extremely useful in compound prioritization, along with the compare type analysis where you have a putative description of the mechanism of action. I think another way we want to improve the process is certainly we'd all like to reduce reagents costs, especially if you're screening quite a few compounds. And if you don't have to include a reporter system in there, then that can be quite useful. But I think more importantly, you want to be able to reduce detection-related assay interference, as we saw with cisplatin. So you don't want your compound interfering with your detection method. And in using stainless methods such as bright field illumination, gives you the opportunity to do that. So these are how we want to improve the process, and this is where we saw the opportunity with the EnSight reader. So I'll just present some preliminary data that we've got using that system. Here's just really an analog of some of the data presented by Norbert, where we were monitoring cell proliferation using bright field illumination versus hours since seeding. This is for an MCF7 line, and you can see that we were able to sort of get a reasonable estimate of confluency. These are our two repeat measurements here in the different colors. So there's and we haven't done an enormous amount of kinetic assessment, but I think there's all the potential there to be able to do that. Looking actually more closely at some dose response profiles, and here we're using colchicine. Colchicine is a compound which affects cell mitosis by inhibiting microtubule polymerization because it actually binds to tubulin. Here we're measuring on the left-hand side. We're just measuring the ATP in the classical method, and you can see that we get an IC50 of around 5 micromolar. When we're using the confluency assessment based upon the bright field image, you can see that we get again an IC50 in the same range as with the cell type glow method. And then using this very interesting parameter which Norbert and his team have been able to pull out of these bright field images, this cell roughness parameter, you can see actually we get quite a nice correlation as well between the cell roughness and the effects on cell viability seen with cell type glow. So I think we're quite interested by that. I mean, what is also quite interesting we've seen consistently is that the cell roughness parameter seems to make the compounds appear more potent, and there may be something happening there whereby cell roughness is an earlier marker for cell cytotoxic effects or effects on cell viability. Okay, we repeated the same set of experiments using colchicine against a renal cancer cell line. Here you can see that the cell type glow assay is giving again around 6 micromolar. Confluency, again, very good correlation there. And again, in this case, the cell roughness parameter gives us about a 3 micromolar IC50 here. So again, there's a good correlation. I think one of the advantages of cell roughness is pretty much orthogonal to both the cell type glow and to the confluency. So you've got a secondary readout of your effect there, which can be extremely useful. And again, here's another cell line, MCF7 breast cancer cell line. Cell type glow gave us close to 10 micromolar. The confluency looked a little bit more potent, but the cell roughness is nearly 4 or 5 times more potent. We think that might be because cell roughness is an earlier indicator. So that's just a view of some of a small amount of the data that we've been generating here, and we're working intensively with the team to extend this to other cell lines. But certainly our initial view of this instrumentation and the opportunity presented by the bright field illumination and the data and the image analysis methodologies is that we get a pretty good correlation between certainly the cell confluency parameter and the potencies that we measure using ATP determination. There's some initial evidence, I think, which shows that extraction of these morphological features, such as cell roughness, will provide a very useful secondary readout. And it may be the case that it gives an earlier perspective on the cytotoxic effects, or it's a more sensitive readout than potentially either the ATP or the cell proliferation. In terms of imaging time, that was less than 4 minutes per plate. I think that's pretty good. That means, you know, it's not the same kind of system as whereby you would have your plate inside an incubator, as some reader systems are. I think you can rapidly read the plate and then put your plate back into the incubator. So there's no unnecessary perturbations to the cell. We have looked at another couple of cell lines, A549 and MDAMB. The data is under evaluation there, but I think initial results show that the actual process of optimizing your image analysis algorithms using the tools provided by the PE4 is pretty straightforward. So we have 35 cell lines that we'd like to get to a point whereby we can do image-based assessment of cytotoxicity of compounds rather than cell type grow methods. So I think that looks likely to be fairly straightforward so far. So the general applicability for these types of cell lines seems to be quite adequate. So where would we like to go next with the EnSight in terms of understanding how it can help with cytotoxicity screening? Certainly we want to extend the image analysis methods to cover additional cancer cell lines, so then we can screen against a larger panel and then see how it is, let's say, in terms of the consistency compared to data which is in the compared database. So we want to be able to do that evaluation. For compounds which have different mechanisms of action, particularly those which are where there's time-dependent features on the onset of cytotoxicity and kinetics, we want to be able to do that. And I think we can do that by with a system. Our system upstairs has full automation and several incubators, etc. And we'll be able to move the cells between the incubator environment onto the plate and read it, and do several reads during that 48-hour incubation process in order to pull out those kinetic effects. We want to be able to profile these compounds which are problematic in ATP determination assays, and those which are affecting cell size and the number of mitochondria within the cells, and that could be extremely interesting as well. And our medium-term goal with this technology is certainly at the beginning to complement, and hopefully in the end we might be able to replace ATP determination with imaging-based assays based upon stain-free bright field illumination. That will allow us to determine cellular proliferation, so that's effects of compounds on growth, and also morphological changes, so for instance roughness, and understand what their impact on cytotoxicity is. At the moment we're not able to unequivocally differentiate between live and dead cells, but we believe that the roughness parameter will give us a handle on that process, and we can better understand that, and that will give us I think a very useful and flexible tool to aid our general cancer drug discovery programs here at Fraunhofer ScreeningPort. So thank you, and I'm just going to move back to Volker, who will give you a summary.
So Phil, thank you very much for your nice and interesting application, and let me summarize before we open the floor for questions. Just a second. So what we have covered in this webinar is what you can do with the imaging capability of the new EnSight Multimode Plate Reader. You have seen and learned how cell imaging can be used for labeled and label-free technologies to get more predictive data earlier in the drug discovery process. How you can improve your data quality, this has been shown by Norbert and by Phil, and how you can reduce cost in cell-based assays by using the cell imaging. And last but not least, how you can use the cell imaging as a nice tool for anti-cancer drug discovery. If you have any questions, want to get more information, I'd like to route you on our website, PerkinElmer.com/EnSight. You'll find a lot of information there. and we're going to populate with more trade shows, events, and application notes and so forth. And now I'd like to open the floor for questions. Please use the Q&A field on your left side. Let me pick up. One question was, what is the field of view? That's a question for Norbert, if you can answer for the different plate formats.
Well, sorry. The field of view is roughly 6.6 millimeter in diameter. So that's really enough for a 384 valve plate and almost enough for any 96 valve plate.
I think the second question is around the same subject. Imaging, the question is, imaging is about 4 minutes per plate. What about the data processing and hit determination? How much time you need to addition for that?
Well, the image evaluation runs in parallel, and for all tests we did run so far, the image evaluation was as fast as the measurement. So the add-on time is just for the evaluation of the last 2 or 3 valves. That's a few seconds only. However, that may of course depend a little bit on the kind of evaluation that is running.
Okay, thank you very much. And now that's next question is for you, Phil. Do you have any experience in monitoring of ATP release over from living cells treated but still inactive cells?
Yeah, I think that's a very good question. I think that's part of our next program of work. We want to pull in compound sets which are, let's say, cytostatic compounds, which will allow us to do that and then do detailed kinetic reads. I think that could be quite an interesting sort of research project, and hopefully we want to be able to do that, and we'll try and publish those results as soon as possible. But that's a good point, and I think having that kinetic information in cells which are not dead completely, but in some kind of quiescent state or affected in some way, understanding what the properties of that population of cells is extremely interesting, and could give us some useful new insights.
Okay, thank you, Phil. We get many questions, so thank you for your active participation. The next is, may I know if the instrument can be used, can be used for suspension cells? That was the question.
Yes, it can. But it's very easy to analyze the cells that do not spread on the floor. What is missing here, of course, is the distinction with the roughness parameter. These cells will always look as very rough, so there is not the rounding up effect that gives additional information on cells that are supposed to die. But except from that, suspension cells are very easy to analyze in bright field.
Okay. Thank you, Norbert. I have another good question. And the question is, can you use label-free plates parallel with imaging? For example, can you use label-free assays suitable for transmission light? I think that's a question for you, Norbert,
yes,
again.
That works really nicely.
So we have initial experiments, right?
Yes, we have experiments, and so very good agreement. We actually run assays, label-free assays, and confirm the effect in bright field imaging after we've run the label-free assay. That works really nicely.
Just an add-on to that, in the experiments that we did, where we had the parallel luminescence readout and the bright field readout, we were able to use the clear bottom plates suitable for the bright field readout and also measure luminescence in there. So you do see, some reduction in your luminescence signal if you wanted to do those in parallel, but certainly the sensitivity of the instrument is sufficient to be able to overcome that. So there's plenty of signal to spare if you wanted to multiplex luminescence and bright field illumination.
Okay, good. Here's another good question. Why don't you have not subcellular resolution on board? That is an easy one to take. I take the one. So the instrument was designed for very fast and easy measurement. That has been the feedback from many customers. We've talked before the design of the instrument, and they see the EnSight as a pre-screening tool later on before they go to high content, first of all, and they like the fact that you don't need special trained personnel and that you get help with space and IT constraints. So that was one of the big arguments. If the application really requires subcellular resolution, I would say please contact our colleagues from the high content portfolio and they can help. I think the main question really is, what is your application? If your application requires subcellular resolution, that's the way to go. If you want to bring simple cell assays, tox assays in combination with multimode detection, then our fast design, our special optical design will help. Good. Hard to pick up. We have so many questions. Is the EnSight based on the Inspire since it used the EPIC technology? Oh, it's for me question. The EPIC technology is exactly the module what you can find on the Inspire. So a simple answer is yes to that question. We go now. That is one, I think, for Norbert. What do you gain by normalization of an assay with cell density?
So from my point of view, the most important aspect of determination of the degree of confluency is to confirm the quality of your plate seeding, of your cell seeding. However, normalization of your assay with the confluency can improve the variation between replicates of an assay, particularly for cases where you have relatively low cell densities. So with 100% confluency, there will be no benefit, but with 20 or 30% confluency, the well-to-well variations can become quite significant, and here normalization does help. We have seen such data in a label-free experiment early on, where we did reduce the error bars by a factor of 3 or 4.
Okay, thank you very much for your answer, Norbert. We're coming close to the end. We have recorded all your questions and answered your questions. Many thanks for your active participation. Thank you very much for joining this morning in Europe and this afternoon in Asia. And thank you very much for your attention. Thank you very much for the two speakers. And just for your information, we have a second webinar scheduled for 6 o'clock p.m. Central European time for our American customers. So if you find out that somebody still is interested in a time conflict, please take the opportunity to join in for our late afternoon call. So thank you very much and have a nice day. Bye-bye.